where we're all coming from. If you could just tell me your name and kind of what your area of interest for research is, then that would be very helpful. Go ahead.
So I'm Rosalind, and I'm interested in making models for nutrient cycling microbial communities and also doing experiments.
My name is Orkun. I work on cellular networks and more recently on microbial communities, especially trying to set up very small model systems and modeling.
I do bacterial and human statistical genetics definitely with an emphasis on inference rather than modeling.
And what was your name?
Daniel.
Phage biology.
Yeah, Forrest and I know each other
Yes,
a little
exactly.
bit.
I'm Jan. I'm from the University of Lausanne in Switzerland, and I'm an experimental microbiologist working in environmental microbiology and bacterial evolution.
I'm Jan. I'm the master student of Rosalind, and so I'm also working on nutrient cycling bacteria, mainly the sulfur cycle.
Paul Joyce. I work with experimental evolutionists, especially those that use a phage model, but interested in, I'm a mathematician by training and statistics, so I do mathematical and statistical models to understand, I guess, evolution and population dynamics through using experimental evolution as a model system.
Great.
Yeah. I'm Christian Pichorano in Delft, and I'm doing biofuel models.
I'm Trishan. I'm from Oregon.
Okay.
I consider myself a theoretician, interested in developing theory that predict rates of microbes in natural environment.
Okay.
I'm Stephen Cornell. I'm from Liverpool in the UK, and I'm an ecologist interested in space parasites and communities.
Great. Okay. Well, it sounds like we have a lot of people who are interested in kind of the smaller scale of microbial interactions, and so that'll be kind of the second or later part of my talk. But to begin with, I'm going to talk about some larger scale issues. So throughout, feel free to interrupt or ask me questions if anything's unclear or you have a thought to share. And just want to acknowledge the funding for some of this research through NSF's Advancing Theory in Biology program and the U.S. Department of Energy Microbial Communities and Carbon Cycling program. So those have been really instrumental in supporting this work. And throughout the talk, I'll name various former students and collaborators who have contributed to some of this work. And the kind of goal for the talk is to show what's happening in terms of microbial processes and microbial controls on soil carbon from the global scale down to very local scales, very small scales of microbial interaction. So trying to integrate across those scales and see if we can come up with better ways to predict what's going to happen to environmental processes in the future as we confront climate change and other environmental change. So you're probably familiar with the important role of microbes in ecosystems, given that most all of us study these microorganisms. But just to refresh, microorganisms are involved in many key ecosystem processes, such as oceanic photosynthesis. And this is an image from NASA showing the distribution of primary production in the ocean, with the greener colors corresponding to higher levels of production. And in the ocean, of course, the main primary producers are actually microbes. They're cyanobacteria primarily. So they're contributing a lot in terms of carbon and oxygen fluxes through the ocean. Microbes, of course, also control processes like biological nitrogen fixation, both cyanobacteria in the ocean as well as symbiotic bacteria in terrestrial systems. In both marine and terrestrial systems, microbes are involved in cycling nutrients, carbon, nitrogen, phosphorus. And in soils and sediments, as well as the water column of the ocean, microbes are key players in carbon sequestration and cycling. And so that's going to be one of the main areas of focus for my talk today. So microbes are really important from an ecosystem standpoint. And most of my background is in soils, and we've been looking at the role of microbial communities in soil processes for the past several years. Soils are a really key component of the Earth system because they contain large amounts of carbon. So greater than 2,000 petagrams globally, which is nearly 3 times the amount in the atmosphere as CO2. And so, for example, here is a picture of a peat bog. This is actually somewhere in the UK. just illustrating that some of these soils can contain massive amounts of carbon. You can visually see it here in this peat profile. And soils, like the rest of the planet, are going to be confronted with environmental change as we move throughout the coming century. And so this is just one simulation from the most recent Intergovernmental Panel on Climate Change report, or IPCC report, which shows the distribution of projected warming across the 21st century, with these hotter colors corresponding to 5 to 7 degrees of warming. So particularly in high latitudes, we're seeing pretty substantial increases in temperature over the land surface as we move towards 2100. So the question that we really have to address is what's going to happen when these pretty substantial climate changes interact with these large pools of carbon that are bound up in soils around the world. So the main tool that we use to address that question are what are called coupled Earth system models. And so these are mathematical models that simulate the interactions between different components of the planet. So these components include things like atmospheric and cloud dynamics, the transfer of energy and momentum and moisture in the atmosphere, and then how that interacts with the land surface and the ocean. So land surface processes include things like vegetation dynamics and soil carbon and biogeochemistry. And then these two components are then coupled with the ocean, which includes circulation and biogeochemistry as well. So it's a massively complex system, right? And these models represent different subcomponents that are then assembled together and coupled in a way that allows us to simulate going forward what the state of the planet in terms of biogeochemistry and climate might look like in 100 years or more from now. So the IPCC, the Intergovernmental Panel on Climate Change, assembles the results from all of the different independent modeling centers that exist around the world, and they do calculations with these Earth system models. And for example, they can generate outputs of what's going to happen to carbon cycling across the coming century. And this is the cumulative uptake of carbon by the ocean and the land surface simulated in these Earth system models. And the kind of lighter colors are the range of variation across these Earth system models, and the dark lines are just the means. And you can see that there's projected to be cumulative uptake by the ocean, which is relatively well constrained. But if we look at the land surface, these models actually vary quite a lot in how much cumulative uptake they predict. So estimates ranging from no uptake, essentially, to relatively large uptake, 400 petagrams, which corresponds to, again, more than half of the carbon that currently exists in the atmosphere. So this tells us there's a lot of uncertainty in our predictions of what might happen to the carbon cycle in the future. Is that carbon going to stay locked up in soils and in vegetation, as these predictions indicate, or could it be released or could it be taken up from the atmosphere, according to those predictions?
And just to give me a feel, is this mostly, I mean, you're talking about in the ocean, usually abiotic things, right? Less of the biology. On land, is it because there's so much more biology that they're thinking about? And
yeah.
Is that the reason?
Right, it's mostly vegetation driven and the interaction between plants and soils. So we'll get into that more in a lot more detail in a minute.
And do the ocean ones have a lot of the biology written into it nowadays, or?
They do actually have, I think, a little bit more advanced depiction of biological processes in the ocean, because phytoplankton are the primary producers there.
Right.
So they're a little bit further along than some of the soil models, as you'll probably guess from my coming slides. So how do we contend with this uncertainty? And I think that these are the questions that have been driving some of my recent research, and doing some analyses on these Earth system models to kind of get a handle on what we might do to reduce that uncertainty. So the first question you have to ask is, how good are the current models? And in particular, how good are they at simulating soil dynamics? Because that's the area of interest for our lab, and because there's so much carbon locked up within that pool. And if they aren't as good as we'd like, how might we improve those models? So what steps might we take to make better predictions? So if you're going to make a very complex model of the Earth system, or even a model of a subcomponent of that system, such as soil dynamics, there are, I think, 4 key steps in the process of building reliable models. And I'll use these as kind of the structure for my talk. And so at the beginning, when a biologist or a geochemist sits down and wants to build a model, he or she has to write down in mathematical terms how the system is assembled and how its components interact. And so this is what I'm calling the process of model development. It's where we take the pools of carbon and the flows between them and couple those mathematically. So that's essentially writing our thought process down into a computational model. So then we have this set of equations, and we need to parameterize those equations, come up with the coefficients that are involved in the equations. And often we can do that by making measurements either in the laboratory or in the field, and we can take those measurements and integrate them into our model framework. And then there's a process of scaling if we're going to look at broader scale processes, not just one place on the planet, but if we're going to scale up to the entire planet or even to an entire country, we want to know how those parameters are going to interact with drivers in the model. So drivers being things like temperature or precipitation, or in this case, soil properties. So those bits of information can be used to interact with the model and scale up its processes across the land surface. And then there's a process of evaluation, hopefully, where we go and see how accurate are the models. Can we compare different models that have different structures? Can we compare the models to data? So maybe we go back to these measurements and see how they are performing. And of course, there can be crosstalk between these different components as well. So doing model evaluation might enable us to improve our parameter estimates if we're using, say, a Bayesian framework or something. And similarly, using better drivers or scaling with a different algorithm might change the way our models perform. So these are the 4 components, and I'll be focusing on each of these throughout the talk. So let's do a little history in terms of the way current models of the soil carbon cycle are formulated. So this is my former graduate student, Kathy Todd Brown, and she did a number of analyses that I'll be showing today. And one of the first things that we had to do was examine how it is that the current models were developed. And you can basically trace back their development to around the 1970s and 1980s. And it turns out that that same structure is still present in the models we use today in 2014. So the basic structure is that we have plant growth and net primary production coming in from photosynthesis. When plants die, they enter the soil, and these models represent various pools of soil carbon. They vary in the number of pools. Some models have only 1. Many models have more than that. They have 3, maybe 4, maybe 10 different pools in which soil carbon can reside. And then we have loss terms, where carbon is coming out of the soil. And that loss process is parameterized as a first-order linear decay process, where the loss is proportional to the amount of carbon that's in the pool and a decay constant, K. And when that material decays, some fraction of it enters the carbon dioxide pool, so the atmospheric pool, and some fraction of it usually goes back into the soil carbon pool or a different pool, which may have a different K parameter. So if we write this as a very simple differential equation, we have inputs from productivity, and we have outputs from what we call heterotrophic decomposition, so the loss of soil carbon. Now, this K parameter is not necessarily truly a constant. It's actually a function. It's a function of our environmental parameters. So parameters like temperature in the soil or moisture, or in some of these models have nutrient dynamics. So those things can influence the value of that decay constant. But the underlying mathematical structure is very similar across these models, and it is represented by this first-order decay process. So essentially what this means is that soil carbon spontaneously decays, right, according to this mathematical form. So we wanted to test whether these current models that we're using to predict the future and make policy, if they're actually a good representation of the world as we know it. So first of all, we actually had to figure out what does the world as we know it look like in terms of soil carbon. It turned out that no one had really quantified an answer to that question. So in collaboration with the Oak Ridge National Lab, Mac Post there, Jim Randerson at UC Irvine, and some other folks, we developed a global map of the distribution of soil carbon.
So, so how deep is the soil?
So that's in the models or in the data?
Well, so in the real data, and in some sense, if it decays in a way, then the further down you go,
The older it gets,
The older it gets.
Yeah. that's what the data show. Now I will
Well,
say,
that's a kind of, yeah. So this is one way of slicing it. You also should slice it down.
although the models actually don't do it that way. They don't have a depth profile. They just have a single column of soil, and then essentially what are different chemical pools represented within that, but that they're essentially assumed to all be mixed together across the depth of the entire soil.
But how deep it actually, so, well, there's the other aspect. How deep, you use sort of 10 meters or 20?
So,
How deep is it before it stops being soil?
well, that varies tremendously from place to place. It could be, essentially no depth down to tens of meters or hundreds of meters even. But this data set, and so we had to account for that. So this data set that we put together counts soil carbon down to a meter of depth. And so there are some issues with that, and that's an assumption that we're making, but that's what we chose to standardize to here.
Okay, but it is pretty close to the surface.
Right, yeah, in general. Although in some of these higher latitude ecosystems with permafrost, there can be a lot of carbon really, really deep in some of these soils. Like that peat bog I showed you, right? That's more than a meter. So again, as you'll see as I go through this part of the talk, there are some issues that are going to crop up. And what I'm showing you here is just the kind of the tip of the iceberg as far as things that we need to consider changing or modifying in some of these Earth system models. But before we can even do that, we have to evaluate how do they compare to the best data that we can put together for the globe. So this is the data set, and there are a lot of assumptions that go into constructing it, but this is just the outcome. And the redder colors are higher densities of soil carbon. And you see that a lot of the areas with high soil carbon are in Alaska, Canada, Scandinavia, Siberia, so the northern latitudes that include biomes like tundra and boreal forest. However, there are also some high areas of soil carbon in the tropics and tropical rainforests, Indonesia, Central Africa, the Amazon, etc. So this is our map that we're going to use as a benchmark, as a comparison for some of the model outputs. So from this map, we can do some additional analyses and calculate, for instance, the amount of carbon that we find in soils in total around the globe. So this is that data set, the Harmonized World Soil Database is what that stands for. This is the total amount of soil carbon. And if we add up all the carbon on that map, it adds up to be about 1,300 petagrams of carbon, which is, again, about twice what's in the atmosphere. Now, the data set actually didn't have a formal quantification of uncertainty when it was put together. So we had to go back ad hoc and do a kind of arm-waving assessment of the uncertainty on that data just to get a range of in our expert opinion, what might be the possible outcomes for soil carbon. So we essentially assigned that range to be 900 to 1,700 petagrams. And we can also go and count up how much carbon is in each of the individual biomes of the world, tropical rainforests in red, grasslands here in yellow, and the greens are the tundra and boreal forests, the higher latitude ecosystems. And then we can compare that distribution to some of the model outputs as well. So here are the model results. These are about a dozen different Earth system models constructed by independent modeling centers around the world. And you see that there's quite a range in terms of total soil carbon, ranging from about 500 petagrams up to almost 3,000 petagrams, so a factor of 6 in terms of difference. There are some models that actually fall within what we think is the range of possibility from the observations, so that's a good sign. And we can also look at the distribution of carbon within these models. Some of the models have very high amounts of carbon in certain areas, like the high latitudes in this particular model, and others have high amounts of carbon in grasslands in this particular model, much lower amounts in higher latitudes. So there's a lot of variation across models in where the carbon is assigned in the world. So this is kind of a summary, and then we can go back in and look at the individual maps that are generated from the model outputs. So here's the data set that I showed you earlier, and on top I'm going to show you 4 examples of different specific models and their outputs. And up here is the correlation coefficient. If we take a point on the map of the observations and a point on the map from the model, what's the Pearson correlation coefficient between those two? And for this model, this is the one of the US models from NCAR in Boulder, Colorado. And the correlation here is about 0.18, which isn't very good. And you can see overall the map is pretty blue on the top, which means this is one of our low carbon models. It was the one on the far left, the figure I just showed you. There are some features that are consistent with the data. There are higher relatively amounts of carbon in the tropics and up there in some of the high latitude systems. But overall, the correlation is not very good. This is one of the British models, the Hadley Centers model, and its correlation coefficient is a bit better. Still not spectacular, but again, you see some desirable features of carbon in high latitudes and in tropical rainforests. The highest correlation coefficient I think we ever got with any of these analyses was about 0.4. So if you square that to get a rough estimate of variance explained across these maps, it's only about 15%. So not terribly impressive. And some of the models are really off the mark. So this is the Max Planck model. Correlation is really low, and you can see that there are some features that are really, inconsistent with kind of observation. If you just are living in these systems, you know that the American West is not an area of super high soil carbon, and neither is most of the temperate and southerly latitudes of Europe. So this model seems to be positioning soil carbon in a very different way from some of the other models and the data.
Are these models based on the same kind of input? I mean, what makes they're all saying they're all told the same thing about what the conditions in the Midwest are, or they told different things?
They're told different things because they're coupled models. Right? These are the outputs from coupled models. So, and I'll show this in a minute, but the amount of soil carbon is dependent mainly on those inputs and on the drivers that control that K parameter, and then also the intrinsic value, the kind of baseline value of that K parameter. So all those things can be changing, and at this point we're not parsing out which of those things is causing these differences. But yeah, we're not necessarily comparing apples to apples, and we could do that, and you could get the models to agree a lot more, although not perfectly. That's part of the interesting, phase of this analysis. So here's finally the Princeton model, which shows a lot of carbon in the high latitudes. In fact, almost all of it is up there, and a little bit in the Himalayan, the Tibetan plateau. So there is actually a fairly high soil carbon there, but not that high.
You mentioned the regression based on the data that you have to figure out the best fitting model within the number of degrees of freedom that you described. So you said that your K depends upon temperature and a variety of other things, which you measure. So can you, among the class of models that encapsulates, can you find the best fitting model?
Yes, and that's basically what we did. If you go to our paper that I'm showing up here, we did a whole analysis of that, boiling the models down to their essential components and looking at across models and within models what essentially is coming out in that regression is explaining the patterns in soil carbon. And I guess, I can just tell you that now. So the primary production, how the model represents that is an important component of it, probably explaining about half of the differences, and then the other drivers and parameterization is about a quarter of the differences among models. And so there's some unexplained variation, but those are the main factors, as you would expect given the structure of these models, which is pretty simple.
But is the primary production being put in from real data at that point, or is it being predicted?
No. So those are not from real data per se.
Predicted
not as
primary production.
Yeah, right. So yeah, if you take these models and you uncouple them, and you take a good data set on net primary production, which I'll show you how we did this later, but if you take that better data set and then force all the models with that same data set, then they start to agree a lot more. And you can get them to agree with the data better as well.
Well, you've put climate information here, but there's like no biological information.
Well, in the vegetation, there's some biological information. So there are, for instance, different functional groups of plants like evergreen, needle-leaf plants versus deciduous broadleaf plants.
So the models are told that?
They are, some of them, in in various levels of detail, but yeah, they are given information like that. Well, sorry, they're not given that information. They simulate that, but they're given the relationship between climate and a certain vegetation type. So
Okay, but they also tell what in each part of the world what the vegetation type is.
no, they're predicting that. So this is fully coupled model. So they simulate the vegetation that exists there, and they simulate the climate. But there is embedded in the model a relationship between those two factors, right? So if the climate happens to be, temperate and relatively moist, then you might get a temperate rainforest there. But it's not specified directly that, yes, we know there's a temperate rainforest there, let's drive the model with that. Or yes, we know that it's a temperate climate because climate can change, right? And that's what we're trying to simulate here.
So are microbes simulated? So you have plants, you have vegetation, and the vegetation falls on the
You're gonna maybe give away the end of the talk, but
Right, okay.
but no, they're not.
Yeah, okay.
I mean, they are in there as a pool in that brown box. They're in there, but they're not functioning in the way that plants function biologically to drive photosynthesis, let's say.
So plants are predicted, but microbes are not.
That's right.
Microbes are parameter change. Yeah.
Yes, exactly. So here's, just to revisit, if you look at the spatial distributions of carbon across these models, they vary a lot. They vary they don't match the data very well, and they vary amongst each other. So there's not a lot of consensus here, which explains, going back to the beginning, that wide range of outcomes we have for predicted soil carbon in the future. The models are very different. And looking again at those future predictions, we can go to these same models and analyze how they're making future predictions. Now, whether we can rely on those predictions or not is probably in question after I just showed you the way that they simulate the present day. But nonetheless, we did the analysis so we could compare the models in terms of their future predictions. So basically subtracted soil carbon stocks in 2100, or subtracted soil carbon stocks today from those in 2100. So this is the change in soil carbon across that 100-year period. And these are basically the same dozen or so models that we've been looking at, but now they're future projections. So negative numbers are losses from the soil, and positive numbers are gains into the soil. So the first feature you see here is that actually there's a lot of models that don't change a whole lot. And some of these are models that have low carbon to begin with, others are not. But there are a lot of models that, despite massive changes in CO2 concentrations and climate across the 21st century, don't show much of a difference in soil carbon. There are a few models that show some modest losses. So this Japanese model, MyRock, and the Canadian models show some modest losses. Other models show pretty substantial gains in carbon, up to about 300 petagrams, like the Hadley Center model. And that's representing, again, a large fraction of the amount of carbon that's in the atmosphere, and also 10% of the carbon that exists in the soil or more. So that's a massive change in soil carbon storage and sequestration. But then we ask, well, what's driving that change? And based on our simple model structure, you would expect the NPP might be involved. So this is plant production. And you see that there is some explanation here, that the models that have the highest change in net primary production also have some of the greatest storage of carbon across the 21st century. So in these models, based on that mathematical structure, if you shove more carbon into the soil from plants, more carbon will stay in the soils. Now, these projections of NPP mean that it's increasing by more than 50% in some of these models. And that's primarily driven by increases in atmospheric CO2 concentrations. So you give a plant more CO2, it does photosynthesis more, and it produces more biomass. That's the assumption here. Now, I will note, however, that the experiments on which these models are based in terms of their parameters have only been done up to about 550 parts per million of CO2. So these are so-called free air CO2 enrichment experiments where you go out and fumigate a forest or a grassland or some ecosystem with CO2, and you measure the plant growth responses. And those have only been done up to 550 parts per million. In these coupled models, the CO2 concentrations by 2100 could in some cases be up to 900 or 1,000 parts per million. So we're pushing the vegetation component of these models well beyond what we know from field observations. But nonetheless, because they're coupled to the soil, those inputs are going to enter the soil and influence that pool.
So why are there these two outliers which have high plant production but not high soil? Like the
Oh, so that's this part here.
I know this 7 and 8 column. I'm comparing the second graph.
Yeah, so
First graph.
these two, yeah.
Yeah.
So that comes back to this component. So that remember, the inputs are only one half of the equation. The loss terms are regulated by that turnover constant, which is dependent on the temperature change in the model and other factors. So we can just boil it down to look at the change in turnover time. So how fast is that pool cycling as we go through the 21st century. And as you might expect based on biology, all of the soil carbon pools in all these models speed up in their cycling, right? As it gets warmer, the assumption in the model is that exponential decay constant gets larger, which is supposed to capture the metabolic response of soil microbes. So some of the models that have the highest change in turnover time, so they're speeding up the most, also are the ones that are losing carbon overall. So essentially, these two factors are opposing each other, and in some cases the loss term wins, and in some cases the input term wins. But in a lot of cases, they basically just cancel out, and that's why we don't see a change at the soil carbon level. So as a biogeochemist, this is what I would expect. Right? I'd expect as it gets warmer that the soil is going to turn over faster and release more carbon. What I didn't expect was these massive responses in terms of NPP. And so those actually cancel out all of the speed up of carbon cycling in the soil. So I think from a number of perspectives, we can say that these models could be better, right? They don't match the current data. They don't match the current distributions of carbon in the soil. The models don't agree with one another. And when you run them out into the future, they make divergent, qualitatively different predictions about whether carbon is going to be sequestered or lost from the soil. And furthermore, because we were able to mathematically boil down these models to that same very simple structure, and because we can trace their origins all back to the same subset of models in the 1980s, the models are essentially non-independent, right? They're all based on the same kind of fundamental assumptions about how the world works, which is this first-order linear decay process. So that gives us some ideas about how we might improve the models. So because the inputs are important, we might be able to, as was suggested earlier, come up with a better way to represent primary production, and that could improve our soil predictions. Or we could come up with ways to represent the other drivers better, temperature changes, et cetera. We might go out and try to get a better sense of what the parameters are, so we can measure the K values potentially in soils or other parameters in the models. Or we could go back to the drawing board and add new mechanisms to these models, or change the way we think that they're conceived. So this is where the microbiology is going to come in. And this is not a new idea, right? So basically what I've told you is that these current models, these conventional models, do not include microbiology in any meaningful way. But we know, going back to at least the 1920s from Selman-Waxman, that microbes eat soil carbon. They're key players in the loss pathway from soil pools. Qualitatively and quantitatively, soil organic matter depends on the activity of soil microorganisms, which are in their turn influenced by soil conditions, including moisture, oxygen, soil chemistry, the presence of nutrients. And this is just a paraphrase, but we've known that this is important for a long, long time. So the next part of the talk is about how we're trying to incorporate these very old ideas from microbiology into our new models of soil carbon cycling. How can we couple microbial biomass and activity in a more biologically realistic way to soil carbon pools? And once we do that, can we explore the responses to changes in temperature and inputs and see if that gives us a more meaningful or more consistent depiction or prediction of the responses? So going back to that simplified diagram, what we're essentially doing here is taking out the first order constant, the K value, and substituting in there microbiology. where the microbes are still dependent on the same environmental factors like temperature and moisture and nutrients, et cetera, but we're also making them dependent on the soil carbon pool. And when we do that, of course, it makes this equation no longer linear, but it's now a nonlinear loss term. So that will, as you'll see, change the dynamics of the predictions of these models quite a bit. So in some sense, it's a very simple change, but in other ways, it's actually quite a substantial change. So we're going from linear to nonlinear. But in another way, we're not actually increasing the complexity of these models that much. We're not adding a whole bunch of new things. We're just kind of changing the way the arrows are, or the pools are connected to one another. So this is the one part of the talk where I'll discuss scaling a little bit. So we came up with a new model of how these pools should be connected through microbial activity and biomass. And then in this paper, which came out last year, we put that model into a global context, and where we could have inputs from net primary production globally that are acting on a model that has microbial biomass and activity represented in a biologically reasonable way. This model has the loss term influenced by enzyme kinetics, so parameters like Vmax and Km, the Michaelis constant. We have CO2 production or respiration from microorganisms represented in terms of a growth efficiency. So the higher the growth efficiency, the more of carbon that's brought into the microbial biomass stays in the biomass rather than respired as CO2. And we have a microbial turnover term, which is actually similar in some ways to the K values that are in the other models. But in turning over, the microbes are actually directly controlling inputs to other soil carbon pools. So they're building up soil carbon when they die. And then we were able to replicate this kind of structure across depth. So we had to go on in the litter layer, which is the kind of duff that sits on the surface of the soil. We represented an organic horizon, so a shallow soil layer that's organic rich, and a deeper horizon that's dominated by mineral compounds like clay. So then we ran this model and again tried to simulate the present day soil conditions. So this is that same observational data set that I showed earlier, but it's just coded in a different color scheme now from this particular paper. And we compared our new microbial model, which is here, to two of the conventional models, this day-cent model and the CLM4CN model. And you'll see that in this analysis, we were actually able to increase the correlation coefficients in the conventional models quite a bit, beyond what I had showed you earlier with the coupled models. And that was simply by using a better input data set for net primary production. So you can make some improvements to the existing models just by changing their inputs. But if we did that along with changing the structure of the model to represent this microbial coupling, we got an even bigger increase in the model performance. And if you square that, we're talking about explaining about half of the variation in the data now. So we considered that to be a success. It's not perfect yet, but it certainly seems to be some progress. That at least we could represent these biological mechanisms and still recover at least as much of the ability to predict the world as it exists today. But of course, what we're really interested in doing is seeing what's going to happen in the future. So we took this model, the microbial model, and compared it to the conventional model responses to an increase in productivity. So if CO2 concentrations in the atmosphere increase, they fertilize plants. That's going to result in more dead plant material hitting the soil. So you simulate a 20% increase. With the conventional models, as I said, if you stuff more carbon in the soil, more carbon will stay there. But the microbial model initially increases and then it starts to decline. And then it kind of oscillates in a damped oscillation until it equilibrates back where it started. And this is because now the microbial biomass is coupled to the soil carbon. So that as there's more soil carbon coming in, that increases microbial biomass, and then that increased biomass can more effectively draw down the soil carbon pool as those microbes eat that soil carbon. And in these models, the equilibrium amount of soil carbon is actually no longer proportional to inputs. It's proportional to microbial parameters, so things like the kinetic constants, the growth efficiency, and the microbial turnover. So that is a really fundamentally different way of looking at soil carbon cycling, right? That it would actually be decoupled from the amount of input. It has big implications for how soils will respond to things like elevated CO2 in the future. And I don't know for sure, whether this is right or that is right. We're honestly not sure yet. We need to do more analyses to test that. It could be somewhere in the middle here. But the point is, it's a very different behavior than you get from the conventional models. And one that's very easy to explain analytically just by looking at the simplified analytical solutions to these models.
So when these enormous layers of coal were laid down, was that a high carbon dioxide time?
It was, yes. Yes.
Okay. But that happened in specific places.
Right. Well, so yeah, in that, I just saw something recently on this. It had to do with the microbes at the time, which didn't have some of the enzymatic potential to break down wood, and so the conditions were highly anaerobic, and the enzymes that microorganisms use to break down wood and another plant material depend on oxygen. So there was it's sort of a chicken and egg, whether the lack of oxygen prevented those microbes from succeeding or that they couldn't succeed because there was no, or
This is kind of bistable state.
Right. Well, yeah. So, but there's two things going on. But there's the high CO2 atmosphere, but then there's also this different climate and different microorganism community that wasn't able to process that organic matter once it died. So that's why we get coal seams, because organic matter fell into bogs essentially and was not degraded because the normal metabolic pathways that would be used today in anaerobic conditions were not available to microbes at that time.
That seems weird. They think that there were metabolic pathways that didn't exist. I mean, sorry.
Yeah.
That seems very weird, right? 'Cause
Yeah, there's something about there's a suite of oxidative enzymes that are necessary for efficient wood degradation that fungi possess. And it's thought that those pathways were not, at least not as prevalent. But you wouldn't expect them to be, as long as it's anaerobic, even today they don't function.
Yeah.
And so it could just be that the world had a lot more swamps, and the conditions were just different enough that the climate was different enough that made an opportunity for.
Yeah. Doesn't seem like yours seems reasonable, but as you increase that litter layer, right, you're gonna rapidly decrease the ability of the microbes to eat it, because it's gonna keep compressing and you're gonna get less and less oxygen.
Well, so that's true. If you did alter the oxygen conditions, this would totally change. But most of the carbon is not necessarily in that state, or the ecosystems are not in a state that's oxygen limited. It should be more so, and that's one of the flaws with both of these types of models, is that they don't do a great job of representing things like wetlands and permafrost. So,
but there are places where carbon is now being laid down.
yeah, yeah, peatlands, sure. Throughout the Holocene, since the ice, the glaciers retreated, it's been accumulating in tundra and other wetlands around, even tropical wetlands. Now, whether that will continue or not is an open question, but thus far there is still accumulation. So yeah, if you take away oxygen, that's a big factor that affects these lost pathways. But again, like I said, that's not represented really at all in any of these models, which is another fundamental problem. But people are working to change that. So that's important.
Could we have a quick look at that model slide with the microbial, this one, yeah. So I guess I had two questions on this. So do you then account for the CO2 that goes out and update your input parameters?
No.
Okay.
So that we're decoupling from the vegetation here.
Because that then perhaps will get rid of that effect that you're saying. I mean, here you're basically creating a sink through the microbial community, that you say the more carbon we have, the faster we're going to degrade it and it's going to go nowhere. Whereas actually it is going into the atmosphere again and it will help more vegetation growth. So it's actually a feedback, but you're not modeling it as a feedback.
Well, but remember that if the pool here is at steady state, then the atmospheric pool will also equilibrate.
But as it stands, the linkage is broken, right, in the model.
Right.
Or CO2 is not creating more vegetation, whereas it should, I guess. And then maybe it reaches a different steady state.
Right. But we're not, but even if you did couple them, I think you would still,
You still get the same effect because you just would expand the biomass even more.
yeah, I think, because we'd have to have a net input of,
But the biomass is modeled as a saturating function.
what's unrealistic is these two, because that's just adding carbon.
And not accounting. No, but I don't know. Maybe this is a longer discussion. But
Yeah.
I'm just throwing that question out. The other question I had, I think I agreed. So you assume that everything goes to CO2, but that's sort of complete fermentation in presence of oxygen.
Well, we don't know. We don't assume everything goes to CO2 because we have this turnover pathway. So some of it goes to CO2, but some of it remains in dead microbial biomass. But there's no other gaseous loss pathway besides CO2.
But also, you will get sort of incomplete fermentation in most of the soils, which will be inhibitory. So, for example, in very low oxygen conditions, it's known if you change the CN ratio, then the community breaks down. And it's not like more carbon. The more carbon you add, the more they just happily chunk it away.
Right. Yeah.
Which I guess will again take away that effect potentially.
Potentially.
We see that there is no change in the soil carbon levels.
Yeah. So there's a lot of ways that, and again, we're not totally comfortable with that strict equilibration.
Right.
So there probably are other pathways and other actually ways to formulate the microbial model that won't give you that same result, and could incorporate any number of additional mechanisms that would not give you that exact result. And so I think that's valuable to do, and we're working on some of those models as well. But the point is that you can't even consider fermentation or incomplete fermentation in a conventional model that we're using today because it doesn't have any metabolic pathways.
No, I'm in complete agreement that this is an improvement.
The degree of improvement is still up for debate, but it's more that we've opened up this Pandora's box of conceptual advances that we can now start to make. Okay, so we also explored the response to warming, since we're interested in climate feedbacks. And so this is the change in soil carbon. This is over 2100. And the conventional models in this case are showing a modest loss of carbon from the soil in this particular parameterization that we used. What we see with the microbial model is a much broader range of outcomes, which depend on the microbial physiological parameters. So in some outcomes, there's actually no change in soil carbon across the 21st century. And in that set of model simulations, we're allowing growth efficiency to respond to temperature. And it's responding in a way that's bad for the microbes, right? We're assuming that growth efficiency is declining as temperature increases. If growth efficiency declines, then respiratory processes increase, but microbial biomass goes down. And since biomass is the main coupled driver of soil carbon turnover, then that carbon doesn't turn over quite as fast. And despite the kinetic effect of the warming, the decline in biomass results in a stabilization of soil carbon. In contrast, if we assume that microbial biomass is very adaptive, that it's able to maintain its growth efficiency even as temperatures get higher, then that biomass is relatively larger in the model, and it can drive down the soil carbon even further. So we got a kinetic effect plus a maintenance of the biomass, and that results in much larger losses of carbon, up to 300 petagrams in this particular simulation. So the outcomes of these models then that are based on microbial coupling, of course, depend very strongly on microbial parameters.
That's what we see in the ocean with
Yeah.
that. Yeah. the worst case scenario.
Right.
basically.
So it's actually, it's better, a better case scenario if the microbes suffer, because then they can't do their job of breaking down soil carbon, and it gets sequestered. Other parameters are probably also sensitive to temperature. This is a new study from some collaborators at Northern Arizona University, and they measured the turnover rate of the microbial biomass as a function of temperature. This is in lab incubations, but they used a bog soil from Minnesota, both organic fractions and mineral fractions, and showed this big increase in turnover with temperature. So the microbial biomass is dying much more rapidly at higher temperatures. They're not exactly sure why. It might have something to do with viral dynamics. It might have something to do with invertebrate predators in the soil. It could have something to do with other internal pathways in the microbial biomass. But if you take this result at face value and implement it in these models, you get some interesting results, which are actually quite similar to the results if you allow microbial growth efficiency to decline with increasing temperature. So here's kind of a control model scenario where we have constant turnover and constant growth efficiency. And again, if those two parameters are constant, then there's no change in microbial biomass. There's nothing here. But there is a change in soil carbon because now the biomass is stable and the kinetic effects take over and we lose soil carbon. If we have constant turnover and declining microbial growth efficiency, this is again the scenario I just talked about, microbial biomass declines and soil carbon actually increases slightly in this simulation. The effect is even more extreme if we kill off the microbes faster as it gets warmer. There's even more carbon storage in this case. So again, this is actually a negative feedback to carbon-climate coupling. So it's really key to know for carbon balance what these parameters are. So we can do measurements, or we can also, I think, use some of the measurements and theory that's been developed over the decades. And there's a lot of it in microbiology, and Steve Frank is coming here next week, so you can ask him about this framework some more, but it's based on many, many decades of research in microbiology. So microbiologists have studied factors that control growth efficiency, and one of the key factors is the rate of resource use among microbes. So the faster a microbe wants to obtain resources, the more it has to invest in acquisition machinery. So more machinery results in more biochemical costs, increasing maintenance, respiration, and energy allocated to the synthesis and maintenance of these biochemical compounds. So that reduces growth efficiency. Whereas if a microbe is more conservative and uses resources at a slow rate, then it can have much less biochemical machinery and higher growth efficiency. This is the otherwise known as the rate yield trade-off. And if microbes are using complex substrates and trying to use those quickly, then that requires an even greater investment in extracellular enzymes that go out into the soil and break down compounds that are too large to take up directly. So we can take this trade-off and impose on it the effect, or the assumed effect of temperature increase on growth efficiency. So if we assume that warming reduces growth efficiency and increases respiration, we might expect that microbes respond to that by moving along this trade-off surface. So they could partly compensate for the warming effect by reducing their investment in resource acquisition. So I've been working on some models that try to implement that kind of a trade-off. And I did a simple analytical model, but I also wanted to show the results from this more complex community model because it gives some slightly different results. So this is called the decomposition model of enzymatic traits. And the key here is that it represents, in a kind of statistical sense, the relationship between microbial traits, so the trade-offs that might occur, like that growth yield trade-off I just showed you. It's a spatially explicit agent-based model with up to hundreds of microbial taxa that are again represented, the diversity is kind of represented in a statistical way, so we're not parameterizing each microbe individually. And it has inputs from plant production coming into soil pools. And the plant chemistry is actually somewhat diverse, so we've got different chemical substrates interacting with the microbes. And we have various microbial metabolic processes, like production of extracellular enzymes, catalysis of polymer degradation, uptake of the monomers, respiration processes, enzyme turnover, diffusion, reproduction, and movement of microbes across the grid. This is all represented in a simulation framework. And the process here that results is the decomposition, the decay of that soil carbon. So you can think about this as occurring on the surface of a piece of dead plant leaf or on a soil aggregate or something like that. So I asked, what happens when we impose 5 degrees of warming on this system? And are there any changes in the what I'm calling the average or intrinsic microbial growth efficiency for the entire microbial community? So that would be the growth efficiency you measure if you measured every microbe at a constant temperature. So do they move along that trade-off surface? So here are some of the outputs from that simulation, and this is several years of simulation, over 10 years of simulation. The dynamics of the different types of microbes in the model. So in the bluer colors, we have the high resource acquisition rate, low efficiency strategy. And in the red, we have the more conservative strategy with low rates of resource acquisition, but much higher growth efficiencies. And there's kind of a mix in the baseline simulation at 15 degrees C. If we crank up the temperature, we do see what I had predicted from that conceptual diagram, that some of the bluer colors are lost, and we see a community-wide shift in intrinsic growth efficiency of plus 0.023, so a 2.3% increase in community-wide efficiency. And we can do multiple replicate simulations and calculate an average and test that against the controls. And so there is a significant increase in intrinsic growth efficiency, but it's actually quite small. Averaged across simulations only about 1.2%. There's actually not a great, very strong potential for adaptation. It's not necessarily evolutionary adaptation. We're talking about a community-wide process here. And if you compare that to the change that's induced by the warming effect, that's actually 0.08 units of efficiency. So it's a small fraction of that. And if you compare it back to the simple analytical model, which is just based on kind of a homogeneous community, that maximum potential for adaptations, half of the warming-induced change. So we just solved that model. And so something about the community dynamics is limiting that adaptive response to be less than what you'd predict based on a very simple system. And my hypothesis about what that is relates to the interactions between microbes in this spatially explicit context. So this is just an example of 5 different microbial types from the model over the 5,000-day simulation. And you can see some of the types drop out over time, other ones proliferate. And for instance, with this bacterial type here, you can see these vertical gaps. And that's where the bacteria are competing with fungi. And they're competing for resources or something. There can also be facilitative or synergistic interactions among these types as well. So there are pretty strong coupled interactions between the different microbial types in this model. And so what that tells us is that in order to lose a low efficiency type, you might also be losing the high efficiency types that are coupled with it, limiting the potential for the community-wide adaptation. So I think that representing the dynamics in a more realistic spatial way can actually generate more accurate predictions of this potential for adaptation. Again, needs empirical support, but an interesting hypothesis from the simulation model. So then just to go back and try to scale up that set of responses and revisit here, if microbial turnover and growth efficiency are constant with warming, remember we're going to be in this high carbon loss scenario, right? Because microbial biomass is going to be maintained in the face of warming. Whereas if the turnover increases or the growth efficiency declines with warming, we're in a much rosier scenario where carbon is maintained in the soil. And the simulation results actually suggest that we might be able to be in that range, at least in terms of growth efficiency, because it seems there's limited potential in a complex community to adapt to that change in temperature. So these microbes might be suffering as the climate gets warmer, according to this limited set of simulations. But again, I think we can extend those types of approaches and look at all the different factors that might be influencing these microbial parameters, and then start to hone this range more and more as we try to scale up these models. So then to just conclude, oh, I guess I am done, huh? The global models differ from each other and from the data, right? So there's a lot of room for improvement there. The conventional models differ fundamentally in their behaviors and their predictions from the new microbial models we're developing. And so I think that there are some distinct advantages to the microbial models, in that they're representing the dynamics more realistically, but there's also challenges because we need to measure now this whole new set of parameters that we don't have the advantage of 30 years of measurements to necessarily back up, or at least we have to go and collect and assemble all of those data. So, but I think if we can do that, then we stand a much better chance of making, or some chance of making accurate predictions going forward. So that's what I've been up to, and I guess we're out of time. So maybe we can chat over beer some more if you have more questions. Thanks.